It is my great pleasure to introduce Professor Ian Winship. Dr. Winship is a professor and former Alberta Innovates Health Solutions Scholar in the Department of Psychiatry at the University of Alberta. He is the director of the Neurochemical Research Unit, and he has his bachelor's in psychology from U of A, his PhD from U of A, his postdoctoral fellow from UBC. So he did eventually leave Alberta for a little bit, and then he came back to Alberta. So as you can tell, he really likes it here, which I can appreciate. So he was the previous deputy director of NMHI, which I obviously keep screwing up the name of. And he is the current associate vice provost of research and development and services. And the title of his talk is Chondroitin Sulfate Proteoglycans as Regulators of Neuroplasticity and Behavior.
Awesome. Thank you very much. How's the mic? Is it good? Perfect. Okay, so thank you very much for inviting me. I think the Glycomics Institute is a real burgeoning success story, so I'm excited to be here and present today and to talk about some work that I don't actually get to talk about that often, which is about the role of chondroitin sulfate proteoglycans in the nervous system. And I'm going to talk about two very different aspects. So historically in neuroscience, to be honest, most of what we've tried to do is get rid of CSPGs. So they are perceived as a barrier to neuroplasticity. And so a lot of work in neuronal injury or in brain injury in different areas of the nervous system, we're trying to get rid of them. And so we think that, okay, the CSPGs are blocking axonal growth, and if we get rid of them, we can allow the brain to rewire and recover better. And so I will talk about some of my work in this area that's doing just that. But I will also begrudgingly admit that there likely is a role for CSPGs in the brain, given that they're highly conserved and they're everywhere. And so I'm actually going to talk about how CSPGs are important for complex behaviors such as cognition and how a loss of PNN can actually lead to cognitive impairment. Okay, so like all principal investigators, I'm going to talk a lot about what we do. And I think most of you know that what we do is actually what the really talented people in my lab do. So I'm going to talk about work that goes back to 2 days ago to 10 years ago. And so I didn't have a single picture that could capture my group, but I wanted to highlight these 5 individuals in particular. So on the brain injury or stroke-induced injury and the role of CSPGs as a barrier to recovery, I'm going to talk about work from Anna Wiersma, a former PhD student, Somnath Gupta, who's actually defending his PhD 1 week from today, and Ron Burtonshaw, who's a master's student in the lab right now. And then I'm going to change directions, and I'm going to talk about cognition and cognitive impairment and the role of CSPGs. And there I'm going to be talking about the work of a former PhD student, Wes Paler, and a current PhD student, Hossein Dusti. Okay, so this is a generic structure for a chondroitin sulfate proteoglycan. This is one flavor. This is aggrecan. They're named according to the core protein. And they all have a kind of similar structure. They have a hyaluronan backbone connected with a linking protein. And then they have their protein core, and then the glycoaminoglycan chains. So keratin sulfate and chondroitin sulfate 1 and 2 on aggrecan. And this is kind of the business end of the CSPG when we talk about this inhibitory role in neuroplasticity. So this interacts with receptors on other cells, including other neurons and growth cones associated with axonal growth and stop that growth. So this is a barrier to the brain's ability to rewire. And of course, they have in the developing brain, they have a role in guiding development through this. It's fitting here. So M and A refer to cleavage points for matrix metalloproteinases and aggrecanase. And I included this particular one because it highlights all the different ways that it can be cleaved, or different places it can be cleaved by those enzymes, because that really has been the focus. And that was how my lab got their interest in CSPGs, was in trying to get rid of the GAG chains and therefore reduce the activity. Now, the reason we want to do that is it's known that in CNS injury, you get an upregulation of CSPGs around the lesion site. So this is an example of a spinal cord injury. So here's the lesion area here, and then surrounding it, there's reactive astrocytes, activated microglia, and then an upregulation of CSPGs. And so a lot of work, especially in the spinal cord injury field, has looked at how we can degrade the skeletal scar to allow axons to grow better through this area. So to reduce the inhibition and allow rewiring. More recently, we've moved into the stroke field, and I'll be talking about that research today. So the good news is that there are established agents. If you're trying to get rid of this inhibitory capacity, there are established agents you can do this with. One of the most commonly studied is chondroitinase ABC, which is a bacterial enzyme, which effectively cleaves the GAG chains from the CSPG, and in doing so, it reduces this inhibitory activity. So there's been a lot of work, particularly in spinal cord injury, that has used chondroitin ABC in different forms to grade CSPGs, and has shown that it does increase axonal growth. It allows axons to grow better through glial scar areas, and it can improve recovery, again, in animal models at this point, though there are some moving towards some human trials for spinal cord injury. Now, this is interesting to us, and in my lab, a major focus has been the relationship between the plasticity of the nervous system and recovery from stroke. So in the event of a stroke, you have a reduction of blood flow that causes an injury in your brain. And if it impacts a motor region, then you have some degree of sensory motor impairment. And so the idea in the field for a long time has been that if you look at motor function versus plasticity of the nervous system, when you have your stroke, if it affects a motor area, you have a severe motor impairment. The idea has been that there's a finite window of neuroplasticity. So a point at which there's gene programming such that the brain is more plastic. It can rewire, it can form new connections, and it can replace some of what is lost due to this injury. But the window for plasticity is finite. During the high plasticity, there's recovery, but at the point that plasticity seems to end, this motor recovery also ends, and most survivors of stroke are left with permanent disability. And so one of the interests in my lab is addressing this permanent disability. So rather than focusing early after stroke, wondering about what we can do out here to basically move that motor function up closer to what it might have been for the animal or for the person prior to their stroke. Okay, the other unique thing about my lab has been we've been very interested in the role of the spinal cord. So in the case of stroke, most injury or most strokes occur in the brain, but we're interested in the role of the spinal cord in supporting motor recovery. We know from a number of imaging studies that the integrity of the corticospinal tract, so the connection between the motor cortex and the spinal cord, is crucial in predicting recovery in human stroke patients. So if you have strong integrity, you have good recovery. If you don't have much left of your corticospinal tract, then you tend to have poor recovery. We were interested whether or not there was active plasticity in the spinal cord, and so this is data from a rat model of stroke where we damaged the forelimb sensory motor cortex, then we looked in the cervical spinal cord, and basically we measured plasticity with different markers, and what we found is that just as I talked about in the last slide, there is this finite window for spinal plasticity. There's a period of 2 to 3 weeks where the spinal cord seems to have elevated axonal rewiring. Okay, so this would be an optimal period for replacing lost connections, but by 4 weeks after stroke, it seems to end. So it is a finite window for plasticity. And if we look at recovery, so in these rats, we looked at skilled reaching. So we damaged the forelimb cortex, then we can look at how well they can perform a reaching task. After the stroke, they're very impaired. They recover during the period of high plasticity, but once plasticity seems to end, so does recovery. So there seems to be this link between finite window for plasticity and finite recovery. And the important thing to know is even if we continue to train these animals, so we give them rehabilitation like a human stroke patient would receive, they don't get better. So the window for plasticity seems to be crucial. We began to wonder, well, what could be closing this window for plasticity, so to speak? And for that, we focused on CSPGs initially. And so this is just some preliminary data, actually some old preliminary data that we haven't followed up on, looking at a couple markers of CSPGs in the spinal cord. So here we've done what's called Wisteria floribunda agglutinin staining, or WFA staining, to label perineuronal nets. And I'll come back to perineural nets later, but basically they're specialized structures in the extracellular matrix made of CSPGs that surround certain neurons. And so you can use them as a marker of whether or not CSPGs are present. And if we look here during these periods of high plasticity, there's fewer PNNs, but after the closure of the window for plasticity, they are at baseline again. Brevican is one type of CSPG. So this is Western blot data. And again, we see the same pattern where periods of high plasticity, Brevican is low. And then at the point where this window for recovery is closed, brevican is high. So we thought maybe these CSPGs are contributing to the closure of that window. Okay, so now, as I said, the two unique things about my work in the field of CSPGs and stroke recovery are that we want to do two things. First of all, we wanted to target plasticity at the level of the spinal cord. We know that integrity is important. We wanted to know if we could increase plasticity at the level of spinal cord, could we increase connections between the injured motor cortex and the motor effectors and improve recovery? And the second thing is we were interested in chronic stroke. So we didn't want to treat right after stroke. We wanted to look at animals that had already recovered as much as they were going to, then treat and see if we could potentiate that recovery. The idea being, eventually, if you could translate this to people, you could have a person who had their stroke 2 or 3 years ago, still has lingering disability, and there might be something you could do to restore lost function. And so this is work by Anna Wiersma. This was an incredibly labor-intensive project where she trained animals on a variety of behavioral tasks, but I'll focus on this skilled reaching task, so single pellet reaching. She trained the animals till they became very good at it. Then we used a focal stroke model called photothrombosis to lesion the sensory motor cortex, including the forelimb sensory motor cortex, such that they became impaired in this reaching task. And then we followed them for about 2 months, during which we could track their performance in this task. Because we were interested in chronic stroke, we waited 1 month and then, or 4 weeks after stroke, and then we targeted the spinal cord. And we injected chondroitinase ABC to degrade the CSPGs to see if we could restart plasticity. We followed them for another 4 weeks, and then at the end of the experiments, we used neuronal tract tracers to look at the anatomy, to track the connections between the cortex beside the stroke and the spinal cord to see if we could increase those connections. During this timeline, in addition to tracking their performance of the task, some animals received task-specific training. So we trained them in the same task, basically gave them practice or rehabilitation in that task to see how it would affect their recovery with or without treatment. Okay, so the first thing, the question was, could we increase plasticity? So these are using track tracing projections of the corticospinal tract from the injured cortex beside the stroke to the spinal cord. So these are control animals that had an injection of penicillinase, which is a control enzyme that's inactive, and these are the treated animals. And if you look at the heat map, this is the distribution of connections of our experimental group, and you can see quite clearly that chondroitinase did increase the distribution and the number of fibers. So we increased the connectivity between the injured cortex or the cortex beside the injury and the spinal cord. So we increased plasticity even though we didn't treat till a month after stroke. So that was promising. What was interesting, if you looked at the behavior of these animals and you did no rehabilitation, so you didn't give them any training, you just treated, left them for 4 weeks and then tested again, there was very little behavioral benefit. So we could increase plasticity, but we weren't giving those new connections of function. To do that, we needed task-specific training. And that's what's shown here. There's 2 different paradigms where in blue we provided rehabilitation, basically. And so the red line shows our chondroitinase treated animals, again, receiving treatment at 28 days, and black is the controls who received a control injection of an inactive enzyme. This is looking at this single pellet performance, so again, this skilled reaching. And we can see that both groups are impaired after stroke, and they recover for a period of time and then plateau. So within this month, as I've discussed, recovery stops. If we treat with chondroitinase and then we provide rehabilitation, so task-specific training, we see an increase in performance. So we can harness the plasticity that we induced by giving them delayed training. They didn't receive any training in this period. However, if we don't induce plasticity, the benefit is minimal. So you need both plasticity and training. This schematic here is showing with early rehabilitation. So these animals received training starting 3 days after stroke and continuing through the entire design. What we see again is that both groups recover, but plateau. So they perform better than the non-rehabilitated groups, but they still stop getting better before treatment. When we treat with chondroitinase and continue training, there's a big benefit. If we don't give chondroitinase, we don't induce plasticity, further training has no further benefit. Okay, so that's my example of getting rid of CSPGs, and the take-home message would be that we can reinitiate a window for plasticity by targeting the spinal cord and by removing CSPGs or using CSP or chondroitinase to degrade them. However, if you don't combine it with rehabilitation, that increased plasticity doesn't have any effect. Now, around the time that we were doing this work, I was also reading papers like this. So this is a paper in spinal cord injury that talked about the two phases of CSPGs. And what this showed is that if you took a spinal cord injury, and in this case, they had biosynthesis inhibition for CSPGs, but if you prevented an upregulation of CSPGs or production of CSPGs later after injury, it had benefit. The animals, again, kind of like what we showed, were more plastic and they recovered better. But if you did it too early after the injury, then the animals actually got worse because that glial scar that forms has a function, and if you don't have it, there's greater injury. So timing was essential. And that got me thinking about, well, maybe there are better approaches than just getting rid of the functional CSPGs. Maybe we can keep the CSPGs, but interfere with their signaling. And for that, we moved on to consideration of a developmentally regulated growth factor called pleiotrophins. And so pleiotrophins has several functions related to CSPGs that were of interest to us and why we thought it might be a good driver of plasticity after stroke. The first is that pleiotrophins can bind to CSPGs, and when it does that, it prevents the interaction with the receptors that drive their inhibitory activity. So a major receptor that induces growth cone collapse and prevents neuroplasticity after injury is PTP sigma, so receptor type protein tyrosine phosphatase sigma. And when PTN binds to CSPGs, that interaction between PTP sigma and CSPGs is impaired. The other thing pleiotrophins can do is it can actually interact directly with receptors, including PTP sigma. And when it does that, it changes their conformation. And in this case, it reduces the phosphatase activity. And it's the phosphatase activity of that receptor that again, drives this inhibition of neuroplasticity. So pleiotrophins can bind and reduce the phosphatase activity. Now, one of the things that's dephosphorylated by PTP sigma is another receptor called the anaplastic lymphoma kinase receptor, or ALK. So it can be dephosphorylated, which reduces its activity, and this is a pro-growth receptor. So this promotes cellular survival and neuroplasticity. So PTN can act on PTP sigma to increase activity, but it can also bind directly to ALK. So it has direct and indirect effects on ALK that can increase this pro-growth signal. So with all of this in mind, we were very interested whether PTN could drive neuroplasticity. Now, so I'm going to talk now about the work from Somnath Gupta in my lab. I can only touch on it briefly given the time. So he's actually defending next Thursday through the Neuroscience and Mental Health Institute. If you get the Synaptic Post, there'll be a link there, and you can learn more about what he's done. But effectively, what he's done is he's used culture and co-culture systems to probe the effects of pleiotrophins on neurons, microglia, OPCs, and astrocytes, but in environments where CSPGs are present. Because what's surprising, if we look in the literature, we know that CSPGs have impacts on those cells. We know that there's less data, but there are several studies that look at the effects of pleiotrophins on these different cells. But what's interesting is very few studies have ever looked at the effects of pleiotrophins on cells grown in a CSPG-rich environment, which is, of course, in the nervous system exactly where they would be. And so that has been the focus of Somnath's PhD. Okay, so I've talked about the ALK receptor and how in indirect and direct ways it might be potentiated by pleiotrophins. And so the first thing we wanted to do was look at whether or not ALK manipulations affected growth of neurons in CSPG-rich environments. And so Somnath did primary neuronal cultures, and he treated with different concentrations of pleiotrophins. So here, this is relative to non-CSPG environments. You can see that the CSPGs here themselves reduce neurite outgrowth, so they reduce the plasticity, the structural plasticity and ability to grow of these neurons. And if we treat with pleiotrophins, there's a significant increase up to a certain concentration. There is a fairly sharp dose-response curve. So pleiotrophins can drive neurite extension in CSPG-rich environments, which is good. And here what we used is an ALK inhibitor called alectinib. ALK has been associated with various forms of cancer, so scientifically that's lucky because there are a number of inhibitors of ALK, one of which is alectinib. And so when we treat with alectinib, this is increasing concentrations of alectinib. We see that even in the presence of PTN, we don't see this neurite extension, this neuronal growth. So this activity of ALK seems to be crucial for the effects of pleiotrophins. Okay, so as I said, Somnath probed a number of different cell types and cell culture conditions. I'm just going to quickly talk about a couple. So one is there is a relationship between pleiotrophine and myelination. So again, in non-CSPG studies, it's been shown that pleiotrophine can increase differentiation of oligodendrocyte precursor cells or progenitor cells into oligodendrocytes. However, this had never been tested in the presence of CSPGs. And that is important because CSPGs inhibit this differentiation. So this is, again, PTGRF alpha is a marker of early progenitors. O4 is a marker of immature, non-myelinating oligodendrocytes, and myelin basic protein would be to identify mature myelinated oligodendrocytes. And what you can see is that in the presence of CSPGs, we don't see mature myelinating oligodendrocytes. So it seems to stop the differentiation. But even in the presence of CSPGs, we can reverse this by treating with pleiotrophine. So pleiotrophine doesn't just act on neurons, it can also allow the differentiation of OPCs into mature oligodendrocytes, even in the presence of this inhibitory signal from CSPGs. So again, that's promising when we think about the in vivo situation. So it can act on neurons, it can act on myelination. He looked at astrocytes, but I'm not going to talk about that data. I will talk briefly about microglia. So one of the interesting things, so he did a number of conditions where he treated pleiotrophine on microglia that were growing in CSPG matrix. So here we have microglia from a CXCR3 mouse, so they express GFP, grown on a CSPG matrix, and the CSPG is labeled in red. And what's interesting is you can clearly see that the microglia seem to carve out a little niche within the CSPGs, which was fascinating. But then what we noticed is when we treated with pleiotrophine, we saw a lot more. So there seems to be more degradation of the CSPGs by microglia when treated with pleiotrophine. And so Somnath quantified that, looked for a mechanism for that. And to do that, he looked at gelzomography and looked at MMP9, so matrix metalloproteinase 9, matrix metalloproteinase 2. These are the active forms of the enzyme which would degrade CSPGs. And indeed, what he saw when we treat pleiotrophine, we see an increase in both these. So it seems like in these microglia, if you treat with pleiotrophine, they actually degrade the CSPG matrix. And again, thinking about neuroplasticity, that would be a benefit. So pleiotrophine can act on microglia as well to drive neuroplasticity. The other thing that was interesting in pleiotrophine that I won't be able to talk about is that the actions on microglia, whether it drove them towards inflammation or towards an anti-inflammatory phenotype, actually depended on whether or not they were pre-activated. So if you come to Somnath's talk, you can learn that pleiotrophine might actually be beneficial both early after an injury, because it moves the microglia towards a phenotype where they're more likely to degrade debris and clear out harmful components near the injury site. But then later on, when they're less activated, pleiotrophine may actually increase neuroplasticity through this mechanism, but also through releasing or reducing the release of inflammatory cytokines. Okay, and so I should look at my time. I just wanted to really quickly mention some new work as we move in vivo. We want to take this work in vivo, but we really need to understand, because pleiotrophine has so many actions on so many different cells, that really does vary by whether they're co-cultured or cultured alone and different conditions. We want to start to get an idea of what happens in vivo. So we started with a really basic bulk transcriptomic study done by Ron Burnshaw. When I say basic, it's not basic to him. It's an incredible amount of work, but the concept was very simple in that we gave a stroke. 24 hours after the stroke, we treated with pleiotrophine around the injury site, and then we took out the brain and did RNA-seq on it. I won't go through the data because I have hundreds of these types of graphs. It tends to drive a pro-growth phenotype, which is really exciting for us. That's what we wanted to see. It actually is active when we inject it into the intact mouse. But relevant to this, I wanted to highlight some gene expression for heparan sulfate proteoglycans. So CSPGs are generally considered inhibitory to growth and migration in the CNS. HSPGs, on the other hand, are promoters of neuroplasticity or plasticity in the central nervous system. And when we treat brain tissue with pleiotrophine, we see significant upregulation of several HSPGs, including syndecan, syndecan2, or syndecan1, 2, 3, and glypican2. And all of these have been associated with pro-growth phenotypes. So again, multiple mechanisms of action, including manipulation of other proteoglycans. Okay, so from those two talks, I've talked about stroke as an injury and the ability to target either signaling or the CSPGs themselves to increase plasticity. And I guess the take home would be that, yes, you can degrade CSPGs and it'll reopen a window for plasticity that can be used to improve recovery. But it also might be worth considering other targets and other ways of interfering with CSPG signaling that doesn't involve removing them. And the second part of my talk, I'm going to talk about why removing them might be a bad thing. Okay, so for this part, I'm going to focus on those specialized extracellular matrix structures called perineural nets that I mentioned. So these are composed of CSPGs, primarily aggrecan, and they are basically, Wes, who did a lot of our initial work on PNNs, calls them like a t-shirt for the cell, in that they cover the cell body and the proximal processes, but they don't cover all of the processes. They don't go all the way down the dendrite or all the way over the axons. And so they serve a protective function for certain neurons. They tend to be primarily on parvalbumin positive or parvalbumin expressing inhibitory interneurons, which are essential inhibitory interneurons for regulating excitation in the brain. And that's where most of our PNNs tend to be found. So there's quite a bit of literature on PNNs. It's known that they're developmentally regulated. So an upregulation in both PV cells and perineural nets is associated with the closure of critical periods in development. So these are periods of very high plasticity. And when these two things show up, that plasticity seems to end. And they're also known to be important for synaptic stability in the adult. So in the adult, we don't necessarily want our synapses changing a lot because they're stable and functional. And they serve a protective function in that they reduce oxidative stress. And that's really important for PV neurons because these are highly metabolic neurons. They have a high firing rate and produce a lot of reactive oxygen species as a result. Okay, and I'm interested in PNNs as it relates to schizophrenia. And the reason for that is that PNNs have a normal development course, which is very interesting because they don't actually develop fully until we're adults. So if you look in an infant, in the cortex in this case, there's very few perineural nets. They start to develop in childhood, in adolescence, there are more of them, but it isn't fully developed till adulthood. So you don't have a full complement of perineural nets until adulthood. And actually the developmental rate will vary between different cortical regions. And they are found in the subcortex as well. Now, what was interesting to us is that this period where they're developing tends to coincide with the age of onset for most individuals with schizophrenia, this young adulthood and adolescent stage. And in fact, if you look in adults, in postmortem tissue from people who have been diagnosed with schizophrenia, so this is a control individual, this is a person diagnosed with schizophrenia, and this is looking in the prefrontal cortex, so an area essential for cognition, we see that there's a significant reduction in perineural nets. And this has been reported for a number of different areas. But of course, postmortem tissue is difficult. In this study, the patients were on average about 55 years. So those in this population of people diagnosed with schizophrenia probably have been on antipsychotic medication for 40 years by this point and had a host of other different experiences in their life that could affect things like PNNs. So we wanted to know whether or not we could replicate this in an animal model. And to that, we looked at a phenomenon associated with maternal immune activation in schizophrenia. So we know from epidemiological studies in humans that in cases where there is, say, a COVID outbreak or a severe flu epidemic, mothers who are pregnant at certain stages of their pregnancy who become quite ill, it affects the baby, and those offspring, those children have an elevated risk of developing schizophrenia later in life. So there's an association between this maternal immune activation and a risk for schizophrenia later in life. And this has been shown for many different illnesses as well as for other stressors. So stressful events like civil wars and stuff can also have a similar increase in risk. So this maternal immune activation can lead to a greater risk of schizophrenia. And we can model this in animal models. So you can take a mouse or a rat, a dam, so a pregnant mouse or rat, and at about gestational day 15, you can treat them with a viral mimetic. So poly-IC is the one I'm going to talk about here. The mouse gets sick for a couple of days, shows sickness behavior, then recovers. The offspring are born at the normal time, and they appear normal size, normal litter size, all that kind of stuff when they're born. If you follow those offspring and then do behavioral testing in them later in young adulthood, we start to see a phenotype that's consistent with schizophrenia. Of course, now this is rat and mouse schizophrenia. We can't tell if they're hallucinating or paranoid, but we can look at things like cognitive impairment. And so when we talk about schizophrenia, I guess I didn't introduce it. Most people think of positive symptoms like hallucinations, and then negative symptoms, which are the more depressive phenotype. Those are very difficult to measure in a rodent. But increasingly, we're realizing that in individuals with schizophrenia, there's severe cognitive impairment. And that cognitive impairment isn't improved by treatment with antipsychotics. So we don't really do anything for it currently, clinically. And it's actually among the most disabling because that cognitive impairment prevents their full integration into society. So these rodents show similar types of cognitive impairment. So impaired social interaction, impaired social intention, impaired recognition memory and spatial memory, and then impaired prepulse inhibition, which is a kind of a measure of sensory gating. So long way of saying we can mimic this maternal immune activation phenotype that happens in humans in rodents. And so our hypothesis was that we could look in rodents with or without maternal immune activation. We could track the normal development of PNNs from infancy through childhood, through adolescence, and then into healthy young adults. And so this is actually rat tissue. And our hypothesis was that we would see a reduction of PNNs in areas relevant to cognition in the offspring of the mothers who were sick. And this is the quantification. So this is looking at prefrontal cortex again. This is the prelimbic region, which is the name of the region in the mouse. And so this is a control animal up here, and this is an animal whose mother received maternal immune activation. And just looking at, you can see that there seems to be a reduction in these perineural nets within this prefrontal cortex region. What was interesting is if we look, so PND7, this would be like infancy, childhood, adolescence, and then postnatal day 90 is like young adulthood for a rat. The PNNs seem to develop normally, but then in adulthood, there's a significantly reduced density and count of PNNs in the poly-IC treated rats in this case. So there is a significant reduction in PNNs, just like we see in the human population. So we were able to replicate this phenomenon of a loss of PNNs in an area relevant for cognition in this animal model. So that was exciting to us, but of course it's correlational. So over the last several years, we've done a lot of work looking at whether or not we can target PNNs, where we can directly degrade them and look at the impacts for complex behaviors like cognition. So this is one of our most recent studies. This is done by Wes as well. And in this case, we used a more sophisticated way of degrading CSVGs. We have an inducible virus. So we can inject the virus into our region of interest. We then feed the animals a doxycycline diet, or not just doxycycline, but doxycycline in their diet. And when they're eating that specialized diet, chondroitinase will be produced in that region. And then when we stop the diet, the chondroitinase stops. And so we did this study in mice. Here I'm showing data where we injected this virus into the retrosplenial cortex. Now, you might not have heard of this, but it's an area that's very important in a variety of forms of learning and memory. So including spatial memory, object recognition, and other tasks. And so we wanted to know, okay, if we degrade these PNNs in the retrosplenial cortex for 30 days, does it have a behavioral effect on measures of cognition? And then what happens if we let them regrow? So they will redevelop. So if we stop this diet, what happens later? So I'm showing data from something called a cross-modal object recognition task. So a basic object recognition task is when you present a rodent with two objects that are the same, you let them explore it, and then you take those away, you delay, and then you present a familiar object and a new object. So that's basic object recognition, and it takes advantage of the fact that rodents will prefer the novel object. So they should, if they remember what they did before and recognize the objects, they should spend more time with a novel object. So cross-modal is a more sophisticated test that requires more cognitive demand and requires the retrosplenial cortex, where you train the animals in the dark. So in this condition, they can go and they can use their whiskers and their paws to explore these objects, but they can't see them. Then in the test phase, you wait an hour, Then you present one familiar object and one new object, but now it's in the light, but they're separated by glass. So they can't use the same sensory modality. That's why it's cross-modal to explore them, and you have to see whether or not they can recognize and spend more time with the novel object. So this is the data over here. So blue are the control animals, red are the doxycycline treated animals. So this is with chondroitinase expressed in retrosplenial cortex. And we can see here that at baseline, both of them, this side here is a preference for the novel object. So both of them prefer to spend time with this novel object. So they've learned, they can tell which one of these from this task is familiar here. However, when we degrade for 30 days in retrosplenial cortex and there's no PNNs or fewer PNNs, which is what's shown here, so a significant reduction of PNNs in the treated animals, these treated animals no longer discern. So they no longer spend more time with one object versus the other, as if they don't recognize them. So they aren't able to do that cross-modal integration, or they don't remember which one they'd seen before. Stop the diet, allow it to recover, and they go back pretty similar to what they were at baseline. So it seems like the presence or absence of PNNs is important. One of the things we didn't expect that we hadn't seen in other studies where we degraded PNNs is that in this study we found that a loss of these PNN structures due to the viral expression in retrosplenial cortex was associated with a significant loss of the number of parvalbumin cells, a reduction in the brightness of parvalbumin labeling, which may suggest that there's less parvalbumin in the cells, and atrophy or shrinkage in the cell body size. So it does seem like a loss of PNNs is affecting the integrity or health of the PV cell population. And this hasn't always been reported in other studies, but most studies didn't use the virus and the sustained administration that we did. The other thing is, the other interesting thing about retrosplenial cortex beyond its role in cognition is that it's heavily enriched in PNN. So if you do WFA staining and you look through the brain, you can see retrosplenial cortex based on the density of the PNNs. So it could be that they're more important in this region perhaps than they are in others. Okay, about 5 or 10 minutes left. So I won't go into this. We did want to look, okay, well, we've shown that there's a behavioral effect of PNN degradation. What happens functionally? So this is work done with Wes and Ryan Zahesi, who's a PhD student in Alan Chan's lab, where they did a kind of regional functional imaging. So they did this in mice that express genetically encoded calcium indicators, and they used a mesoscale imaging approach where you're actually just looking down at the surface of the cortex, and this is over time. So this is basically the earliest image, and then we're collecting images this way, and this is the last one. And you can look at fluctuations in fluorescence intensity of these calcium indicators. And what those reflect are aggregate neuronal spiking. So this is the whole mouse cortex. So this is obviously large regions, and each of these bright spots represents an active area of the brain. So we can look at retrosplenial cortex, which is kind of this area right here, and we can look at spontaneous fluctuations over a period of time at baseline, and in mice either with control treatment or with the doxycycline and the degradation of PNNs. And what we see is there is a significant reduction of activity in the retrosplenial cortex in the treated mice. This is a log-log scale, so if we took this out, it would actually be a very significant reduction in activity. The fact that it's at low frequencies, we have to do, this is in electrophysiology. We don't know whether that's an artifact of the fact that the dyes are slow or whether it actually is low frequency oscillations that are changing. Okay, so the last thing I'm going to talk about for 5 minutes or so is tangentially related in that so we showed that in a developmental model of schizophrenia, we have a loss of PNNs, and that if we target PNNs directly, we can alter brain activity and we can induce cognitive impairment. So that seemed to support the idea that PNNs are involved in cognition and cognitive impairment in schizophrenia. We wanted to now look at a completely different model, so a non-developmental model of schizophrenia that doesn't, to our knowledge, have any direct relation to CSPGs or PNNs, and see if, using that model, we would also see changes in these perineural net structures. And so this is work that Hossein in the lab has been doing over the last several years, and he actually has a poster today where he'll go through this data and more. And what he did is he used this chronic ketamine model. So ketamine administered at a sub-anesthetic dose will induce in rodents a behavioral phenotype that is somewhat, that can be considered psychosis-like. So it's associated with cognitive impairment and other symptoms that are thought to align with positive and negative symptoms in humans. That's acutely. You can also give it chronically. So what Hossein does is he gives a sub-anesthetic dose for 14 days straight to young adult mice, so not during development. And then he lets the drug wash out and begins to test behavior and a number of other things in the weeks that follow. And the reason we're interested in ketamine is because it touches on NMDA receptors, and so this is thought to potentially replicate the glutamate hypothesis of schizophrenia, but it also affects other receptors that can account for different symptoms associated with the disorder. And so it's thought to be a reasonable model, as I said, of psychosis, and we wanted to know, well, if we use this model, do we see changes in perineural nets? So the first thing we had to do is confirm that we had a behavioral phenotype. Again, this is 14 days of ketamine at a sub-anesthetic dose, followed by a few days, and then behavioral testing. This is the more simple version of the novel object recognition task, where they're exposed to two of the same items. Then in a test phase, they have a novel item and a familiar object. And in the controls, you can see that the saline-treated, the non-ketamine treated animals prefer the familiar object, whereas especially in the males, and again, a male dominant phenotype isn't unusual in different models of schizophrenia, and likely reflects some of the epidemiological differences in terms of age of onset and severity of symptoms that we see in males versus females in humans. We see that, again, these male mice in particular do not prefer the familiar object. So whether they don't recognize it or they can't remember what they've seen in the past, they have an impairment in this task. That's consistent with other models of schizophrenia. We also tested spatial memory. So here we use the Barnes maze. And so basically this mouse is dropped in the middle of a maze with visual cues around it, and there is one escape hole. And the lights are on, so this is aversive for a rodent. They want to get out of the light before something comes from the sky and snatches them. So they're going to look for the escape hole. And so over the training phase, we do this, and they start to learn to use the cues to find that escape hole more quickly. Then in the test phase, we take away the hole and we measure how much time they spend in different quadrants to understand how well they've really learned where that escape hole was. And you can also do other things like measuring working memory and errors, but I won't talk about that today. So this is the data here, again, for male and for female mice that were treated either with saline or with ketamine in gold. And in both sexes, in this case, we can see a significant impairment. So they spent less time in the correct area. They were potentially less sure of their memory and explored other areas rather than focusing on where they knew the escape hole was in the past. Although I'm anthropomorphizing there, so I can't actually tell you what the mouse was thinking, but they don't perform the task as well. Okay, so that was really interesting to us, but again, we wanted to come back to PNNs. And so we looked in retrosplenial cortex again, because it's important for both those tasks. We know this from other studies. And we saw two interesting things. So first, we looked at microglial cell counts by staining for IBA1. And we see that with this ketamine treatment, there seems to be increased inflammation. Down here. So more of these microglia. And again, remembering that microglia can release things like matrix metalloproteinases that can degrade PNNs. Then we looked at PNNs, again, using WFA, and that's shown here. And again, we saw a significant reduction. So this adult pharmacological model of cognitive impairment in schizophrenia seems to impact perineural nets. And we see cognitive impairment similar to what we see when we directly degrade perineural nets. So again, supporting the idea that this might be a core feature of schizophrenia across different causes. Okay, and so the last thing I'm going to talk about is some really early work. So we're lucky in my lab to have a 2-photon microscope, so we do a lot of in vivo microscopy. One of the things we can do is we can use transgenic mice that express those genetically encoded calcium indicators that I mentioned before, and we can zoom in on a region of brain and we can image neural activity in a group of neurons, and we can do this in anesthetized mice or in awake behaving mice. So mice that are head fixed, but on a platform that moves with them, so they think they're moving. And so what Hossein has been doing is using that system to image the activity of mice with or without chronic ketamine. And so this is kind of what it looks for here. So they're in a floating platform, but they're head fixed to the microscope. So it shows them moving, but actually the platform is moving. And this guy's sitting here checking some out. When he moves, they're not synced. You'll see the activity. Now he's going to hang out here for a while. They're not playing at the same space, so you can watch this guy, and then you have time to go over here because it's delayed slightly. But when he moves again to a new environment, you'll see a surge of activity in these neurons. Right now. And so we can do this, and we're starting to incorporate simple behavioral tasks in this as well. But what Hossein did is look at activity in awake and animals that are moving or aren't moving in retrospinal cortex with or without ketamine. So this is, so as I said, they're head fixed, but this is the platform moves around with them so they can check out the outside. They do also move through the inside. It's not like they're stuck on the outside. And we can look at the activity when they're moving. That's what's tracked here. And you can see many of the neurons in this area were synchronized, but you also see activity in some neurons when they're at rest as well. And so this is very preliminary data. This is about 2 animals per group, or for 2 males and 2 females per group. But we do seem to see that this chronic ketamine seems to alter neuronal activity. Again, primarily in retrospinal cortex, it seems to be in males. But again, this is very early, so I wouldn't bet on this in the long term. That said, so this is non-PV neurons. So this is neurons that don't express parvalbumin, and this is the population of parvalbumin expressing neurons. We do double crosses so that we can get a reporter for PV neurons in the same animals that we have the GCAMs in. And what we see is that without ketamine treatment reduces the activity of the non-PV neurons, which should be primarily excitatory, but increases the activity of inhibitory neurons. So it does seem like chronic ketamine and potentially the loss of PNNs is affecting the activity of these cells. And so Hossein's going to be following up on that over the next year before he also finishes. So thank you, everyone. That's my talk for today. So again, in that last section, hopefully what I've shown is that this loss of PNN seems to be integral to schizophrenia, and it can be replicated in different models and is associated with functional effects. And so happy to take any questions. Before I do, I tried to highlight Wes, Somnath, Hossein, Anna, who did the bulk of the work that was presented. Yongli and Trisha are my technicians, and they're the reason the labs functions on a day-to-day basis. Catherine Todd co-supervises Somnath and is a great collaborator. And then Alan Chan, Kareem Fouad, Glenn Jickling, and John Hallen at the University of Saskatchewan have all made significant contributions to the work. And of course, our funding sources, we've been lucky enough to be funded by a variety of sources that allow us to do the work. So thank you.
Questions?
Thank you so much. Very interesting on multiple different levels, but I think I followed much of it. Did I follow correctly that in some cases the female response was amplified and in others it was suppressed? Did I follow that correctly? And if so, can you comment a little bit on some of the differences? In is it a model specific thing? Is it a treatment specific thing? Or do you think it's a real sex difference?
That is a really good question. Yeah, so we quite often, so this is relative to the schizophrenia work, quite often you see a stronger phenotype in males, but it's not universal. So that's a general statement across models and across cognitive tests. Now, I don't think I presented any data where we did the same cognitive test, unfortunately. Because we've moved to some more sophisticated things like cross-modal for a lot of our stuff. So the answer, I suppose, would be there are real sex differences in the presentation of the disease in humans and in mice, according to and there's a different immune response in the case of maternal immune reaction. So there are real sex differences that are in the literature. In our case, focusing on PNNs, we have not detected in the animal models a difference in perineural net loss between males and females, but we're not always necessarily powered to do that. We do always do both sexes, but we don't always have the power to say. And it hasn't been reported clinically. That said, in some of our tests where we degrade, we do see a stronger effect in the males or females. And at this point, we haven't got far enough to say whether that's a true sex difference or just an artifact of the test that we did.
Very interesting talk. Sorry, I was late. So
No
back
problem.
there. My question is regarding the poly-IC.
Mhm.
That so did you try an infection model, because poly-IC is very strong? So did you try like a bacterial infection to see that you can see the same phenotype?
Yeah, we have not. So that work, the actual poly-IC work was done by John Howland in Saskatchewan. But other groups have used other mimetics, other ways to induce maternal immunoactivation. And they do see different degrees of phenotype, but I would say overall, I think the timing is more important than the actual maternal immune activation. So across the different ways that you can induce the maternal immune activation, the offspring do tend to have a phenotype. It might be stronger with poly-IC than others, but it's more the timing of when you do it that's crucial.
So the follow-up question, have you done or anybody has done anything to see that if it's through the development, fetal development, or biologically the components following hyperimmune activation or chronic inflammation, if there are milk components, so do you know by fostering like pups, to see that if it's through milk, there are any components that can kind of cause or replicate similar pattern?
I don't know about the answer to that specific question. Certainly, our big question when we look at the rat data is the actual deficit in PNNs we see is about 100 days after the intervention. And up until that point, and this is an immuno study, so it's quite crude, we don't see differences, very few differences in any of the markers we looked at for other things, including PNNs, but also looking at PV cells, total neuronal count, astrocytes. We don't see overt inflammation at that stage. So we don't seem to, again, very coarse measure, just looking at IBA1. And other groups, surprisingly, it hasn't been investigated. Like I don't know that there's a consensus yet whether this is a developmental effect that changes the trajectory of genetic programming that doesn't occur until 90 days or 60 days or whenever it actually occurs, or whether or not it's a sustained inflammation. But certainly there doesn't seem to be anything overt in that model in our case. That said, in human autopsy, they do tend to see things like increased IBA1 cell counts that do suggest there's some sustained inflammation. But again, in postmortem tissue, it's difficult to say whether that was happening when they developed disease or whether it's an artifact of the many other things in that individual's life that could be, be a stressor. So I don't know that the answer is no.
I was wondering, I don't know whether I got this right. So you were showing that when you do the treatment with the chondroitin, can't remember the
Chondroitinase, yeah.
Yeah, with chondroitinase, you get a reduction in the number of parvalbumin neurons. Is that correct? That correlates with decreased cognitive capabilities.
Yeah.
And then you can reverse at least the cognitive effects. So the two related questions are, does the perineural network actually, the lack of perineural network induce cell death in these neurons? And if that is the case, the recovery, do you think that actually is there a migration and differentiation of progenitor cells in that area? How do you explain these things?
It's a really good, it's a really good question. I think, so the one thing that we've been careful of is we think that it's a loss of PV cells, that there is a loss. So it's with the PNN degradation. Important to note that we only saw that when we did it for 30 days with the virus. When we just inject the enzyme, it has a shorter duration of activity. And we haven't seen significant loss of PV cells. So we have analyzed, and it seems like there actually is a loss of PV cells, but we can't actually prove it because they could still be there, but we don't see the parvalbumin. So it could be a reduction in parvalbumin expression. That said, I did show the data where we looked at intensity. It seems like there's a reduction in PV, but so we counted faint cells as well, but we still think overall there probably is a loss. And we think it affects PV cells because they're highly metabolic, and one of the roles of the PNNs is oxidative stress. So we do think there's injury. Now, we have looked at 30 days and 60 days. I think I've showed most of our tissue was from 60 days, and now we're going back and looking right at the point we end the treatment, because we want to go back and look and see if we can see active markers of cell death or cell injury, because we didn't look at in the 60-day tissue. We still see that there's a loss or a lower count of PV cells, but we can't look at ongoing injury at that point. Does that answer your question?
Yeah, so basically there is a connection also to the second part of the question of the potential regeneration, or is it more rewiring?
So it could be both. If there's a loss of neurons, so we haven't ourselves tested for neural stem cells, but we do know that degradation of CSPGs is associated with increased migration. So you should see migration of neural stem cells and others into the area if there's enough of an injury signal to draw them there. That said, at least in injury models, we tend to see very few of these actually stay as neurons. So they're more likely to turn into glial cells or other things. So I would think in that case, because we're looking at, like, it's not a lesion site, I would think it's probably more about unmasking of existing connections to replace function, stabilization of synapses that might be affected by the loss of PNN, but aren't degraded and that kind of thing, as opposed to like an actual neurogenesis process. Yeah.
Yes, a question at the back.
Thank you for the wonderful talk. I have just a curiosity whether the perineural network, is there a flavor of aggregation of CSPGs in other type of highly metabolic cells in other organs? Is it something like a common strategy, or is only occurring in the brain?
That's a really good question that highlights my bias as a neuroscientist, because I actually don't know the answer. But I mean, there are other highly metabolic neurons in the nervous system that also don't have PNNs. But then at the same time, it's not only parvalbumin neurons, they are on other neurons. So, I mean, I think the answer is, with respect to PNNs, we don't know entirely why they select the neurons they do. The best rationale is that PV neurons are highly active, but not all PV neurons, only about 70% have a PNN. So there's still this other population that's somehow different. And I actually don't know. It's a really good question if we looked in other organs and the most metabolic cells, and if you would see an enrichment of CSPGs around them.
So,
I just have a quick question. So I saw in one of the NMHI talks how the use of certain recreational drugs can lead to lower the age threshold for the presentation of schizophrenia. So do you know if that can also change the PNNs?
It's a really good question. Again, those studies haven't been done. We are working with John Howland actually on a project looking at maternal cannabis use and how that affects things, including PNNs, but other things. But I don't know whether if you look at that adolescent at-risk group, which is associated with increased risk, I don't know whether it affects their PNNs. And I don't think anybody's looked at whether cannabis directly affects CSPGs or PNNs at all. There's also a big debate about whether it's actually just accelerating the onset or actually inducing schizophrenia. And so depending on who you talk to, it's fairly controversial. But yeah, I don't think it's been looked at.
One more question was, are there different flavors of a CSPG? I would imagine so. And is there a switch actually from when it's protective or versus when it's detrimental? What do we know about actually the different forms of CSPG?
What do we know, or what do I know? No, yeah, I do approach this from a neuroscience perspective, which is why I highlight that initially I was mostly interested in getting rid of them. There are many different CSPGs. So I mentioned, so the very first one I showed was aggrecan. They're defined or named according to their core protein. So there's a number of different CSPGs that have different core proteins and then different glycosaminoglycan chains. Now, those chains are dynamic. They can be larger or smaller. They can be sulfated in different ways that affects their activity. So basically, it's very complex. And yes, the answer is yes, there is a switch that will affect the action of the CSPG, depending on, and so aggrecan is found as an example in PNNs. It's the major constituent of perineural nets, but it's also found in the interstitial matrix. And I would predict if you could isolate the two forms and put them in a dish and test them, you would see that they would have different effects on neurons. One likely more protective, one more likely, stopping growth.
Other questions? If not, I have a ton, but I think I'm going to save mine for when, for when we're not waiting for lunch. Please join me in thanking Professor Winship for an amazing talk.